Hello, this is Beverly Chester, and thank you for coming to my presentation on necrotizing fasciitis. Just an interesting tidbit on why I chose necrotizing fasciitis, because 20 years ago in high school, I did a project for anatomy class and chose necrotizing fasciitis as my topic, and that's ultimately what got me so interested in human anatomy and healthcare. So I feel like I am bringing my project full circle. The clinical trial that I found is called Detection and Delineation of Necrotizing Fasciitis via a vascular perfusion fluorophore. The purpose is ultimately to determine if this diagnostic test can be developed using the indocyanine green solution as a fluorescence signal, and to see if with imaging, are there any intensity changes associated with necrotizing fasciitis and vascular perfusion. It is a prospective cohort study with an estimated 15 participants, all aged 18 years and older. The study start date was April 5th, 2021, and completion date is estimated at April 4th, 2025. The location is in Lebanon, New Hampshire. The eligibility criteria are adults aged 18 years and older with clinical suspicion of necrotizing fasciitis, with one or more of the following findings from a physician or mid-level provider assessment. And the findings are pain out of proportion to visible findings, a temperature of 100.4 degrees Fahrenheit or higher, the LRINEC score of greater than 6, and that is a diagnostic tool used specific to necrotizing fasciitis. And if there are tense but soft tissues. The adult could be determined to be a candidate for soft tissue biopsy and or surgical debridement by the on-call emergency department, orthopedic, and or general surgical trauma teams. Finally, the adult should have the ability to give written informed consent. Exclusion criteria includes people with a history of allergy to the indocyanine green and or iodine in general, pregnant women or nursing mothers, and those who have no comparator region available for imaging control. The primary outcome measures will be the diagnosis of necrotizing fasciitis using the indocyanine green, from here out referred to as ICG. The administration of ICG and the subsequent imaging of a bodily region affected with necrotizing fasciitis will demonstrate substantially reduced fluorescence compared to the patient's unaffected tissues. Demonstration that ICG fluorescence voids are characteristic of necrotizing fasciitis, from here out referred to as NF. There are no stated secondary measures. In summary, necrotizing fasciitis, also called flesh-eating bacteria, has a high mortality rate that leads to rapid soft tissue infection to a patient's subcutaneous fat and the fascial layer, which spreads centrally to a patient's entire body, leading to systemic sepsis, multi-organ failure, and death. There is no definitive diagnostic test for necrotizing fasciitis. The current method of detection results in non-specific diagnostic lab results that would indicate a number of non-fatal conditions, leading to the underdiagnosis or misdiagnosis of necrotizing fasciitis. Back in high school, I learned that Jim Henson, the creator of the Muppets, died from necrotizing fasciitis in my home state of North Carolina because he was misdiagnosed as having the flu. Again, these diagnostic lab results would indicate a number of non-fatal conditions, and his non-fatal condition was considered the flu, and he was sent home and he passed away. ICG is an FDA-approved substance with excellent safety record and no demonstrable toxicity. ICG paired with fluorescence imaging maps out vasculature, allowing the viewer to distinguish regions of perfused and non-perfused blood flow. The prothrombotic effects of a necrotizing fasciitis infection lead to the hypothesis that administering ICG and imaging the bodily region affected will demonstrate reduced fluorescence compared to the same patient's unaffected tissues in the controlled comparator region. Data management for this project would include internal data, the diagnosis of necrotizing fasciitis, imaging results in a patient's chart, the diagnosis of suspected NF in a patient's chart, and biometric measurements from a patient's chart. External data could include previous historical records through the Centers for Medicare and Medicaid, state and federal health departments that might have previous data, historical controls of NF, and NF state case studies or other reports. Primary data sources will be the actual patients with clinical suspicion of necrotizing fasciitis. Secondary data sources could include previous NF case studies, clinical trial registries also being performed for necrotizing fasciitis, and clinical research papers. To maintain data integrity, we would train all data users. Data footprints for all users would be utilized so that any changes made by one data user could be easily tracked back. We would ensure all notes are transcribed as is from patient records, no editorializing. We would ask for more clarification from clinicians when needed. All users of data would save their own version of their manipulated data file, and we would have a single master file that would incorporate any user data manipulations. And again, partnered with their data footprints, we would be able to go back and find out which data manipulated which user manipulated which data. Some example of data analytics could be ordinal values given to disease severity, and the example I derived was number of 1 through 4 for low severity, 5 through 7 for moderate, or 7 to 10 for severe. And we could group similar groups of disease severity. That way we could study similar groups and dissimilar groups. Numerical values could be provided for the amount of vascular perfusion or lack thereof. For example, we could use percentage of perfusion, maybe have the like groups grouped for how much vascular perfusion has been diminished. Let's say 25% has been diminished in one study group, and we could group those like perfusion groups together and study them versus a study group that has 50% or greater perfusion, for example. We could also include imaging photos with the explanations of conclusions that were drawn from the fluorescence imaging. And implications to health outcomes would include the individual health outcomes are likely to stay constant if nothing changes, or it could result in better treatment for these ICG imaging groups than with control groups. Either the ICG and imaging will produce quicker identification of necrotizing fasciitis to produce better outcomes for that patient compared to historical controls, or it will not, resulting in no difference from the control. Ultimately, the ICG and imaging could produce better health outcomes. That's the hope and the purpose of the study. And so many patients with clinical suspicion of necrotizing fasciitis could be helped in the future. The references I have used are here, and the clinical trial is the reference from the United States National Library of Medicine. Thank you for coming to my presentation.